Today, we'll be learning about the creation of computational pipelines and how they help researchers go from a mess of jumbled genetic information to a simple genetic test that breeders and veterinarians use to improve the health of horses. Genetic research has played an essential role in the advancement of human medicine and has similarly played an important role in animal medicine. The creation of genetic tests for horses allows veterinarians to accurately treat their patients and breeders to avoid mating pairs that would perpetuate a disease. Current technology has made it possible to sequence all 2.7 billion nucleotides found in the horse genome. Manually evaluating every single nucleotide is a physical impossibility, with reading a single genome taking the average human over 9 years. Computational pipelines will take hundreds of genomes and rapidly analyze them. This is why the creation of computational pipelines has become so important. This is the outline of a basic genetic pipeline. The first step in this pipeline is to align the DNA sequence of the subject horse to a well-established reference sequence, rebuilding a horse's complete genome from the many chopped-up pieces of DNA that sequencing technology creates. After alignment to the reference genome, any locations where the subject horse differs from the reference are marked for further investigation. These locations are often known as mutations, but are more commonly described as variants in genetic research. This process is repeated for every single horse in the research population until a large list of variants is created and passed to the next step. The next step of the pipeline is variant prioritization. Variant prioritization determines which variants detected in the previous step are most likely to cause disease. A pipeline can be created to include multiple methods of prioritization, but this one focuses on the use of variant frequency analysis and variant effect prediction. The first step in determining if a variant is disease-causing is examining the prevalence of each variant within the diseased population of horses and comparing it to the prevalence in the healthy population of horses. A variant that occurs in all or most of the diseased horses, but rarely occurs in healthy horses, is a good candidate for a disease variant. A variant that only occurs in a few diseased horses is not a good candidate, as it cannot explain all cases of the disease. A variant that occurs in equal amount in diseased horses and healthy horses would also not be a good candidate, as it is found in too many horses that do not have the disease of interest. The second step in determining if a variant is disease-causing is variant effect prediction. In this step, the pipeline uses several tools to predict the effect a variant has on the function or regulation of a protein. A variant can affect the function of a protein in many ways. The worst-case scenario is a variant that prevents a protein from being created at all, leaving its function unfulfilled. A variant could also cause the protein to misfold into an incorrect shape, preventing it from binding to receptors, substrates, or other proteins, which may render the protein unusable or simply cause it to be less effective. Another subset of disease-causing variants can also be found in areas of the genome responsible for regulating the production of proteins. These variants may cause too much or too little of a protein to be created, disrupting the fragile balance of cellular function. Using the variant frequency analysis and variant effect predictions, the pipeline can assign scores to each variant, indicating how likely they are to cause the disease of interest. The pipeline will give a short list of variants and their associated scores that researchers can examine further using a larger population of horses or functional experiments. This pipeline has been built to reduce the amount of time and labor necessary to find the single variant out of millions that is responsible for causing a disease. The pipeline allows for researchers to standardize their methods and complete studies larger than anything possible without computer processing, pushing the field of genetics forward and improving the welfare of horses everywhere.